Hello everyone, welcome to yet another class of learning with online biology tutorials. I hope you are liking my content. If you want a change, you can leave a comment in the comment box, or even if you have any problem, you can leave a comment in the comment box. You can drop me an email, you can leave a message on my Facebook page and on my Instagram ID. I'm active there too. So in today's lesson, we are going to start DNA replication. Now, as I said in the very first video of this chapter, that the flow of information, it is going to happen from DNA to RNA to proteins, right? So the DNA which is already there in the cell, it will get replicated, right? Now, replication occurs whenever cell division has to occur, and this we have seen in grade 11. So, very important is when it occurs, okay? There are 3 to 5 questions which I would be telling you in the end, and you are required to prepare them all, right? So, question can come in any form, just stay tuned till the end. So DNA replication, when does it occur? Now you know that cell can divide, and generally there are 2 divisions through which a cell divides. One is mitosis and the other is meiosis, right? Mitosis happens in the somatic cells and meiosis happens in the germinal cells when reproductive cells or gametes, they are to be formed, okay? Prior to any kind of cell division, there is one stage which is known as interphase that has to happen, and then only the cell will divide either by mitosis or by meiosis. Now, when interphase is happening, we have also seen in grade 11 that generally there are 3 phases in the interphase, and it starts with G1, S phase and G2 phase. G here stands for gap, S here stands for synthesis. So when does DNA synthesis happens? DNA synthesis happens in the S phase of interphase. This you have to learn, because for 1 marker question you may get when does it occur. So whenever a cell is destined to divide, prior to it interphase occurs, and in the interphase there are 3 substages, G1, S and G2. DNA replication happens in the S phase of the interphase. So whenever we want to form a cell, right, these are the stages. Like whenever cell has to form from the pre-existing cell, then interphase has to happen and then cell division has to happen. So one thing we have already learned, right? Now we will see how DNA replication occurs, right? DNA replication occurs because it utilizes variety of enzymes, and one amongst its category is the DNA polymerase. DNA polymerase is a one which would be helping in DNA replication. There are different types of enzymes for DNA replication in prokaryotes and eukaryotes. In today's video, we are speaking in general for the DNA replication, okay? Now DNA polymerase would be the one that will be helping out in the replication process. Simultaneously, there are other DNA polymerases as well, which will help in the proofreading and the repairing that will be done during the DNA replication. Rather, DNA polymerase first, that is meant to destine for the synthesis, it will do the synthesis. Then later the other polymerases will come and proofread. Is everything right? Are the bases complementary to each other? Are they attached as per the base pairing rule? If there is any error, then the DNA polymerases that are meant for repairing, they will make the corrections. Because you know, if any error will be left behind. It will lead to mutations, and these mutations could be of varied type. Even cancer is the reason of one of the mutations, so we will be talking about in the further videos. So DNA polymerases are of variety. Some will be synthesizing the DNA and another will be doing the proofreading and the repairing, right? So we will be discussing the DNA replication here in the steps. So let's get started with the steps. What would be the steps? Okay, so first thing is that origin of a replication will be decided. Origin of replication means the point from where a replication has to start. It will be defined. And remember, DNA replication, it is going to be bidirectional and semi-conservative. How it is semi-conservative, I have already explained in the Michelson and Stahl experiment. So if you haven't watched that video yet, please do so. So origin of replication means the point from where DNA replication has to start. Now DNA has certain autonomic replicating sequences, right? These are autonomic replicating sequences. In abbreviated form, they are known as ARS. These ARS are the sequences with specific bases. Once they come, the ORI site is defined. This ORI means abbreviation of origin of replication. So once ORI site is defined, means this is the point where the replication has to start. Now the second point would be these bases like adenine, cytosine, thymine, guanine, they will be activated, right? So what happens is these bases, they are generally in the nucleotide form, right? But they will be in the monophosphate form, like deoxyadenine monophosphate, deoxycytosine monophosphate, deoxyguanine monophosphate, deoxythymine monophosphate. Here small d means deoxyribose because the sugar in DNA is deoxyribose. These are the bases, 4 bases, and MP means monophosphate. So all these nucleotides, they will be converted, they will be activated into the triphosphate form, right? So DCMP will be converted into DCTP, and so as others bases, they would be doing it, right? Once they are activated, then the process, it is going to start. This process, this conversion process requires the energy, it requires some more phosphate molecules, and it also utilizes the enzyme phosphorylase. Okay, these are the necessities for this step to happen. Now the very next step is very important. Here what would be happening, the replication fork, it is going to form. Now replication fork is formed. Now here the role of the enzymes is also very important, which enzymes they are going to help. Okay, so what will happen, first an enzyme helicase will come. Now this helicase will do the unwinding of the DNA. You know, earlier days PNT phones were used, right? And they had a wire that was extremely coiled. DNA resembles such coiled structure, so it is extremely wound, it is extremely coiled, right? So helicase will do the unwinding. Another enzyme, topoisomerase, will come, and what it will do, topoisomerase will remove the strain, because by unwinding of the DNA, strain will be developed, and this strain will be removed by the topoisomerase. In certain books, its name has been mentioned as the gyrase, so don't consider gyrase is another set of enzyme. So topoisomerase and gyrase, they are altogether same, right? The another thing which you need to learn are the single stranded binding proteins. So here SS means single stranded binding proteins, right? We will be seeing them all in the double helical DNA. Let's see first and then we will move further. You know that this is the coding strand and this is our template strand, right? DNA is double helical. So I have just shown you a piece of it. Now, as this DNA has to open, ORI site has already been defined, and we will open like this. So you can see here a fork-like structure is there. This is our handle and the front part is resembling a fork. Okay. So this double helical DNA is opened. This is our 5 to 3 prime and this is our 3 to 5 prime. If you will not show arrows at 3 prime here, it will not be counted as mistake, but it is always better to show. Right? Now, helicase would be the one that will do the unwounding. So this is our helicase. And as I said, topoisomerases, they will be binding and they will help in removing the strain from both the strands. Okay. Now, these single stranded binding proteins are the ones. What they will do, they will keep these strands as single stranded, because these strands, they would be acting as a template for the synthesis of the new strands. Okay, so this green colored structure is the topoisomerase. This blue colored structure is our helicase, and the black colored structures in the diagram which I have shown you, these are our single stranded binding proteins. Make sure that you label each part of the replication fork quite correctly, because question comes, draw the replication fork and label it. Okay, so till this step, you need to learn that the replication fork will be synthesized. Means every step has been done now. What has to happen? It will be happening in the next step. So replication fork is synthesized, is done, right? DNA, double helical DNA has been opened. Helicases, they have done the unwinding. Topoisomerases, they will remove the strain, and single stranded binding proteins, what they will do, they will keep it single stranded. I promised that I would be asking you a question in the end. So I just wanted to tell you that in the board, they may ask you in which stage DNA replication happens. They may also ask you to draw the diagram of the replication fork, which is ultimately important. So in any case, you will not leave it. Rest DNA replication, few steps are still left which are very major. So initial steps are done. Rest we will be covering in the another video. Thank you and stay tuned for part number 2 of the replication.